So what does that really mean? What that really means is that we have to domesticate algae. So we've done exactly that with all of our crops, right? We spent 7,000 years turning teosinte into corn. Teosinte is the direct predecessor, and it's not very many genetic mutations actually to get from here to there. It's only about 10 fundamental transcription factors get you what corn looks like today. But it took a very long time under traditional breeding. The enormous advantage we have now because of the genomic revolution is an ability to do that in a fraction of the time, right? And then that we believe that we can build design-for-purpose crops. So how do we do that? And I'm just going to spend the last couple of minutes going over my street cred details, okay? So you can transform algae. They're eukaryotic, which means you can either put genes into the nuclear genome or you can put them into the chloroplast. Inside the chloroplast genome is where all photosynthesis takes place. Chloroplasts come from photosynthetic bacteria, so they look a lot more like bacteria, except they're inside of a eukaryotic cell, so they have all of the machinery to fold complex proteins. But they have very simple promoters, and the rules are pretty simple. Nuclear gene expression in algae is just like nuclear gene expression anyplace else. Complex promoters, proteins can be transported and moved all around, they can be secreted, you name it, okay? All right, so what have we done? Well, the first thing we did was we made rainbow-colored algae, all right? We actually took fluorescent proteins, we took fluorescent proteins, blue, cerulean, green, yellow, orange, called tomato, red. So what does this have to do with biotechnology? Well, probably nothing, except that I believe that if you can make rainbow-colored algae, you can make almost anything, right? In truth, you can do some very sophisticated things with fluorescent proteins, and this is simply where we went back and labeled the nucleus, we labeled the mitochondria, we labeled the chloroplast, and we labeled the endoplasmic reticulum. So clearly these are very powerful tools, but we can also use these in all of the high throughput screening. All of that high throughput screening that was developed for the biomedical industry absolutely can be applied to algae, and we've done that, okay? Now I just want to show you a couple of the modifications we've made. One is we want to think about fuels. Here's the pathway for beta-carotene biosynthesis. It's an isoprenoid. It comes from a 5-carbon precursor, isopenyl pyrophosphate, converts all the way down to beta-carotene, the precursor of vitamin A. So you can also shuttle these things. You can come in and grab a geranyl pyrophosphate and make monoterpenes. So monoterpenes are C10s, and the one we chose is called limonene. Limonene is the C10 isoprenoid that's found in lemon and lime peels, right? And if you've ever taken one of those and squeezed it into a candle, you know that that stuff burns really well. In fact, it's 138 octane gasoline. So we took the genes for that monoterpene synthase, we introduced those into the algae, and here's accumulation of limonene inside algae. So we have lemon fresh algae now, okay? But what that really shows is that we can do any of the metabolic engineering that you can in every other system. And in fact, here's simply 2 different genes put in, and then accumulation of a C30, which is a direct precursor of fuels, okay? So we can modify fuels any way we want. We can also do much more sophisticated things, and here's one of my favorite ones. These are called immunotoxins. So what immunotoxins are is they have an antibody binding domain, and then they have a toxin domain. So the antibody will bind to any cell you direct it against. We happen to direct these against something called CD22, which is a cell surface antigen on B-cell lymphomas, okay? That antibody binds to the cell, internalizes the toxin, and the toxin kills it, all right? The reason we can make these things in algae, so you cannot make these things in any other system. You can't make them in any other system because bacteria aren't sophisticated enough to fold a complex protein like that, and you can't make them in eukaryotic production systems because the eukaryotic toxin will kill them. We could make these in algae because we sequestered these things inside the chloroplast. And as I said, the chloroplast is a prokaryotic organelle, so it's not susceptible to the same eukaryotic toxins. So we made those guys. This simply shows accumulation of the single chain, the monomeric immunotoxin, and the dimeric immunotoxin. We purified those and showed that we could not only kill B-cell lymphomas. So here's just the antibody alone binding. It doesn't kill the cells. Here's the monomeric guy. It binds and kills the cells. Here's the dimeric guy. It binds and kills 10 times more effective. And we could also use those to treat solid tumors, human implanted tumors in mice. So one of the most sophisticated anti-cancer drugs available on the market. Now, these things are enormously expensive. There's a company called Seattle Genetics that has one of these just coming out of phase 3 clinical trials. That drug will sell for $160,000. So clearly, if we can produce it in a system like this, we think we can impact that price. All right? So that's nice. We can sort of do some of those things, but we really want to talk about, and what I really want to talk about today is what are the sort of the game-changing technologies that we could bring that algae offers us an opportunity. And here's one that I really like. So colostrum is the first protein that's put out by all mammals, all mothers, and what it does is it imparts passive immunity to their infants. All right? And there's a variety of things that come in colostrum, but two really important ones are you get antibodies, and those are antibodies directed against all the antigens that mom has seen. So mom gives those antibodies to her baby to give that baby protection from the same antigen she has seen. And then you also get a lot of different growth factors. And so one of the really interesting growth factors that was identified in there is called mammary-associated amyloid. And what this protein does is when you eat it, when any infant eats it, it goes into the gut, so it's taken orally, gets into the gut, and it stimulates mucus production in your gut. And what that mucus production gives you is protection from bacterial and viral infections, not specific bacteria, but just generally from getting bacterial diarrhea. And many of you will know that bacterial diarrhea is the #1 killer of children on the planet. It's not malaria, it's not starvation, it's dehydration, right? So we got a grant from the Gates Foundation, took this algae, showed that the protein produced inside of the algae was bioactive, and then just the last week, we finished trials in which we fed the algae to pigs and then monitored, their weight gain and their susceptibility to bacterial diarrhea. And in every case of the pigs that ate the algae with the SAA and with the MAA in it, they showed dramatic weight improvement, and they showed loss of bacterial diarrhea. So it seems to work. So what this says is, now we have a protein, a drug essentially, that we can produce, and you have to feed about 1 gram of this. So what's a gram cost? Well, 1 gram of algae, you can produce algae at about $8 a kilogram. So 0.8 cents a gram. So for less than a penny a dose, we now have a drug that can treat bacterial diarrhea. Okay? So the last thing that I want to talk about is, okay, so we can make lots of cool products, and I think there's enormous opportunities for these things. But I preface this whole thing to say that what really is coming and what we really need to think about is how we can become hyper-productive. What does the Green Revolution 2.0 look like? So one of the things we wanted to ask was, could we impact photosynthetic function? And I just want to show you a couple slides that shows we can. So photosynthetic complexes, this is photosystem 2, so this is the complex that splits water into protons and oxygen, one of the toughest reactions on the planet. And this is dictated by and large a set of proteins here called the reaction center proteins, right? And how those proteins behave very much determines the photosynthetic efficiency of the cell. So what we did was we went back and looked at cyanobacteria. So cyanobacteria grow in every environment on the planet. They've adapted really well to low light, high light, you name it. And they do one thing really interesting, and that is that one of their reaction center proteins, called D1, has a low light version form and a high light version form. So if the cyanobacteria grow at the bottom of a pond, they express the low light version form. If they grow at the top of the pond where they get a lot of sunlight, they grow the high light version form. Okay? So what we asked was, could we take those genes from cyanobacteria and introduce those into our green algae and take that gene and get the same phenotype? And what you can see right here is this is actually a measurement of electron flow through the photosystem. And what you can see is the highlight version, whether it's in a cyanobacteria or whether it's in the green algae, has the same electron flow kinetics, and the low light version, whether it's in the cyanobacteria or in the algae, has the same energy kinetics. But more importantly, when we actually grow these things, when we take an algae that we have introduced a cyanobacteria gene into it, what you see is that the low light outcompetes the high light version form when grown in low light. So here's the low light version form, here's the high light version form grown in low light. The low light guy grows quicker, right? The same is true if you do the opposite. If you put the highlight version form in and grow these guys in highlight, the highlight version totally outgrows the low light version. And that's actually because the low light version in highlight photo bleaches and kind of dies. So in other words, we have taken a gene from a very distant relative, introduced it into this algae, and impact its ability to do photosynthesis. So I think what that really tells you is that we can do extremely sophisticated engineering of these organisms, and we can use these things to impact basic processes. Now, as I said, photosystem 2 is actually a very complex reaction center. It's actually got 6 different genes, A, B, C, D, E, F, spread all over the genome. So what we did was we went back in my lab, cut all of these things out, deleted them all out by genetic manipulation, and then reintroduced a synthetic cassette, so a gene that was completely made by de novo synthesis of it, with all of its control elements, its 5' and 3' untranslated elements, the entire thing, and we introduced that back in at a single locus. And when we did that, sure enough, we can get very nice photosynthetic function. All right? So that says that we actually can use synthetic biology, completely synthetic techniques, to impact photosynthetic function inside of these guys. All right, so I'm going to end here with just a couple of final points. Okay, so we can do a lot of fancy things. What's the final frontier? The final frontier in bioenergy is absolutely going to scale. Okay? This is a picture of the Sapphire facility down in Columbus, New Mexico. Each one of these ponds right here is 1 acre, and these ponds over here are 2 acres. They have 100 acres under production today. It's very productive over this summer. They produced about 100 metric tons of biomass, and they converted that into fuel, which has now been converted into jet fuel. All right? But as we develop the technologies to produce bioenergy, as we take algae and put it out into an agricultural setting, get it to really agricultural scale, we have to, this will drive the price down because of the economies of scale. And as we do that for bioenergy, we do it for everything else that we can produce in algae. We do it for food, we do it for therapeutics, we do it for nutraceuticals, we do it for everything. This is brand new agriculture. Importantly, the agriculture and the species that they're growing right here are tolerant of salt water. So these are grown in about 50 millimolar sodium chloride, but there are some species that are marine algae that can grow up to 5 molar sodium chloride. All right? That means we are not impacting fresh water to do this. So this really is new agriculture. And in fact, if we could identify the genes that give us this saltwater tolerance and transfer those to terrestrial crops, we'll completely change the game of food production on this planet. If we had corn that could grow in 10 millimolar sodium chloride, there would be no problem. And rice that could grow in that, there would be no problem with food production on this planet. All right? So I'm going to leave with one last slide, which has already been shown at this meeting. I think Eric Lander showed it on day 1. And what this is, I think this is really interesting. So what this is here's the plot of the cost of sequencing, right? And as he talked about, that price has gone to nothing. So in other words, the cost of sequencing is no longer limiting. In fact, sequencing is now so cheap that we have a collaboration with Life Technology. We make some of the vectors for them that they sell in algae. We send them new algal genomes of algae we isolate, and 2 weeks later, they send us back 16 gigabases of sequence for our bioinformatics guy to assemble. And it is so cheap for them to produce 16 gigabases of DNA that it's not worth filling out the forms to actually charge us to do that. Okay? But here's the other really interesting aspect of that. This is computing power. And there are estimates that computing power will go infinite in the next 5 to 10 years. So what does that mean, go infinite? What that means is that computers can now design the next generation of computer faster than we can actually build it. All right? So the summation of these two things, in my mind, tells me that biological information will very soon not be rate limiting. If I'm a student, I'm thinking about how do I integrate these two things together. It's the bioinformatics, right? All of the experiments that we do in the future are going to be done in silico. We're going to do them on a computer. We already do it that way now in my lab. We look at all the genes that are out there. We predict what their function is, right? The rate limiting step for us now is actually manufacture and testing of the strain. It takes us longer to synthesize the DNA, do the transformations, and isolate the strain to prove that we were right than it does to figure out what the next experiment is to do. So that, in my mind, tells me that the era of genomics is about to become the era of synthetic biology, right? In which we actually start to build design-for-purpose crops. Right now we have fit-for-purpose crops. That means we've selected them to be corn, to be rice, to be all of the other things. But in very short order, we're going to be designing crops specifically to do something. And I think a very important part of that will be algae. I think it could be a very important part of that. All right? So I'll end it there and just say that you can follow us. So I'm the director of the San Diego Center for Algae Biotechnology, and you can follow us there. And we've also started a new research unit at UCSD called Food and Fuel for the 21st Century, in which we are attempting to integrate not only the plant and algal biologist, but the social scientists, the economists, the engineers, all the rest of the groups that have to get together. Because one of the things that we have to face, right, and I've had this discussion with several of you here, is that, you know, we're really good in science about developing new things, about building new strains, about understanding genomes, about understanding how things work, and we're really bad about thinking of what the political and ethical and social implications of those are. And we really need to start to think about that, right? Specifically for me, in making the colostrum proteins, this is a real issue because these things could save literally millions of people worldwide. But if I show up in China, or I show up in Africa, or I show up in Indonesia with a strain of algae and tell these people, yeah, just eat it a little, make you feel better, right? Your kids won't get diarrhea. It's very unlikely that they are going to accept that, right? Yet another, you know, white man medicine brought down to save us. So we need to think about these things, and we need to integrate these things because this era is coming, all right? So I'll leave it there, and I think I have a few minutes for questions.
Questions for Dr. Mayfield? Mike's in the middle and on both sides. In the center, please.
Hi. As you can tell from the weather here, it's hard to grow algae in November.
Yeah.
The Green Revolution 2.0 works well in Southern California, I'm sure.
Yeah.
Where do you see this Green Revolution, the algal revolution, in a cold climate?
Yeah, so, you know, I think a lot of people have this misconception that, you know, that we're going to end up growing algae only in the southwest deserts of the United States because it's warm and there's a lot of sun. This is agriculture, all right? Where is agriculture? Does Canada not do any agriculture? No, of course they do. Do they have different species than we do in the United States or that they do in Mexico? Yeah. You guys don't grow corn here, you grow wheat. It's the same thing with algae. Okay, you won't grow the tropical versions of algae or the ones that are high temperature, but you will absolutely grow algae that are prepared for this environment because it's simply agriculture. Will some places be more productive than others? Yes. You know, if you grow corn in Illinois, you get a much higher yield than you do in Texas, but you still grow corn in Texas, right? And I think this will be no different.
Maybe on the left-hand side, please.
Okay. Hi, I was wondering if you could speak a little bit to the downstream processing of the algae, because one of the big bottlenecks is not just producing the algae, but also extracting the oil or extracting the value-added products. And so if you could speak a little bit of how you would harvest all that algae.
Yeah, so what I said, you know, I was a little bit glib when I said that the problem with algae now is an engineering problem. That is actually exactly what I meant. It's no longer a biological problem. The biomass productivity is high enough, the oil content is good enough. You know, what we really need to develop are low energy. They don't have to be enormously efficient in terms of high throughput. They just have to be low energy input for extraction of the oil and then for reprocessing all the rest of it that is not in the oil. I specifically do not work on those things in my lab, not by choice, it's just because they're an engineering problem. Enormous progress has been made in the last 4 or 5 years. You know, several years ago, people thought, oh, you have to dry the algae and then do hexane extractions because that's the way we get oils out of seed crops. That's a very expensive way to get oil out of anything, right? And people learned very quickly, oh, no, you don't really have to do that. You know, by going to a little bit of high temperature and by altering the pH a little bit, you can actually extract oil from wet algae biomass, no problem at all. So I think a lot of processes are coming along, but that is clearly an area that needs to be improved.
Last question in the middle, please.
I really enjoyed your talk, but I was thinking about what has really changed in North America in terms of fossil fuel production. I really think it's shale gas. I think that's a huge change. And so we see very low prices, and they're here to stay. I mean, there's enough shale gas for a century. They started to build all these plants to bring gas in from outside, and now the gas goes the other way. I mean, so LNG terminals have to be reconfigured to export.
Yeah.
And so that is really, this area here right now apparently imports gas from the United States. It doesn't come from Alberta anymore. You got the Marcellus Shale underlying New York, and production is huge. So I think the competition is not so much $80 to $90 oil. It's $2 to $3 per so many cubic feet natural gas, which seems to be present in an absolutely humongous amount in North America. So it's the only comment.
Yeah, so I realize that wasn't really a question, but let me address the comment there. So the comment is that because of fracking, we now have methane so abundant and so cheap that will become a new energy resource for North America. That is absolutely true, with a couple of important exceptions. One, there's a $14 trillion infrastructure of petroleum refining and gasoline distribution, and there is zero infrastructure for natural gas. So we are going to have to spend that same amount of money to get that going. Two, fracking does not increase the resource that is available. It increases the rate at which we get it out. All right? One of the little things that the gas companies and the oil companies have kind of done a little bit of this on is, oh, look at this great abundant natural gas we have now. You know, instead of 25-year supply, we now get 4 times as much gas. We have a 100-year supply. That is completely wrong. What we have is 4 times the rate of extraction. We haven't increased the resource. We've increased the rate at which we extract it. Natural gas is going to be a large, bolus that is going to go through the system for the next 10 years, not for the next 50 years. This is a 10 or 20-year dividend that we're going to get, and then the price is going to go back up again as the supply goes back down. So in some ways, I think this isn't, and I hope I did not imply that any bioenergy, and specifically algae, is going to be an answer to our energy problem. It's not. It's an all-in equation. We need to do everything. We need to convert as much as we can to natural gas. We need to, get bioenergy and solar and you name it. Because, one thing that has not been discussed much in our country lately, and I think not here too much either, is climate change. Hey, you know what? Turns out that stuff's real, right? And that stuff is going to dramatically impact the price of food, along with the increase of the price of fuel driving that. Climate change is going to impact it too. So anything that we can do that will mitigate that in any way, I think is a good thing. And clearly 100% of the fuel we get out of algae, that was because we sequestered CO2 from the atmosphere.
Thanks very much. Please join me in thanking Dr. Mayfield.
Thank you.
Thank you.